Okay, welcome everybody. Good afternoon. Hello, welcome everybody. I'd like to get started to make sure the students have time for their talks. So please bear with us. Thanks very much for everybody for coming out today. It's a very exciting day. Just had match day a few days ago. That was exciting. I'll have graduation coming up in a while. That will be exciting. Next phase of lives for the graduating seniors, all kinds of exciting places and residencies to go to. That will be exciting. But what's really exciting is the research symposium. So we at the school are extremely proud of all of you students and the incredible work that you do year after year. It never ceases to amaze us. The things that come out of a phenomenally compressed schedule, as you know, and time availability that look a lot like very sophisticated and are research studies you normally attribute to somebody who's dedicating their entire time to a research project or graduate education. And somehow you manage to do that every year and come up with great pieces of scholarship and work, publications, presentations that makes not only you and your mentors proud, but the school and the entire enterprise of the VTC. So this is really a great day that culminates in a recognition of that work. And I think you're all going to see today examples from your classmates of some of the spectacular work that's gone on. And in addition, you've already gone around hopefully and seen some of the posters. We'll see more later to see the rest of the graduating classes' work, including those that do oral presentations today, to show the consistency of the excellence of work. So congratulations to all of you, first of all, for the incredible work that you've done. Today is a celebration of that and a chance to share in that material. So we're going to start off today. I'm sure you've all seen the program, but there's something a little bit different than we've done in the past. This, by the way, is the 5th annual symposium, research symposium, based on the number of classes that have graduated. I thought it was the 8th a minute ago, and Dr. Laconte reminded me we didn't have one the first year when nobody was graduating, so thank you for clarifying that for me. Anyway, we're going to do something a little different, and we're going to take some time at the outset to recognize a couple of the mentors that you have worked with. And so I think, as all of you know, we had an opportunity for people to weigh in and give us input to try to decide who were some of the really terrific mentors that you've been working with over the past few years. We sent out questionnaires and surveys to graduates and those of you in the school and received a number of those back. And we decided to recognize 2 mentors for their outstanding service this year to you, the medical students. And so it's going to be my honor and privilege today to present them to you and say a little bit about them. So we first had a selection criteria that I think you're all familiar with. We asked for people to evaluate if these mentors create an environment in which original hypothesis-driven research is effectively implemented and guided, if they provide opportunities for professional growth in their research operation, if they challenge you to make original research contributions and carry out your research to completion and with rigor that your patients and all of us expect of you for this research. to foster intellectual growth and technical methodological proficiency, to establish clear expectations and maintain high standards for quality, ethics, and work ethic. And so we got back nominations and suggestions and comments, and I'm going to share a little bit with you of the comments about the first one of the honorees that we're going to mention today. I'm just quoting from some of the letters that you all and your colleagues sent in. They said, this person marvelously balances teaching medical students with other responsibilities as a principal investigator in an effort to encourage us to be independent thinkers by stressing technical proficiency and autonomy. However, what we all admire the most of his approach with us when our experiment fails. Always an email click or a door knock away, takes time out of his day to listen to our rationale, carefully comb through our results, and provide us with guidance on how to proceed further. His approach has taught us not only how to deal with failure, but also how to spin a less than ideal situation into an opportunity for learning and growth. Others said he has worked hard to instill in us a strong work ethic, passion for research, and pride in our growing abilities. He's an incredibly caring and inspiring mentor. So it is my pleasure to bring to the podium the first recipient of the Mentors Award, Dr. Shang from the VTCRI. And I'm sorry, before you say a word, I did want to mention one other thing. I had it in my notes here. Xi has mentored 10 medical students so far. He's graduated 4, has 6 in his lab currently, a few about to graduate, and 4 of his students have won National Foundation Awards for the research they've been doing with him. So Xi, would say a few words. Go ahead.
Okay, thank you. Thank you, Mike, and thank you all the students for this terrific award. And I think this is not just for me, it's also for you guys. And this is also a special appreciation to all the students who dedicated themselves in working in my lab. I told Mike that I have 10 students, but he probably didn't know why I'm accepting so many students to my lab. This is the reason. So more students could nominate me for this award. Every time when I talk to Mike, to Leslie, to Jen, how great, how fantastic the medical students in my lab, I was not exaggerating. I actually meant it. They are terrific. They are outstanding students. I have many stories to share, try to share with you, but today I'm going to just share one with you. It happened like a few years ago. I got a crazy idea at night. It's almost midnight, but I was so crazy about this idea, I couldn't wait to tell everyone in my lab. So I sent an email, told them the idea and how to prove this idea. And just a few minutes later, I got a reply from Pratik. And he told me he was interested in this idea. And then he spent a few weeks testing this idea. And he put everything together. And he published this paper in 2016 when he was just an M2 student. He is terrific and outstanding. And you're going to hear this data today. He probably will talk about it. And finally, I want to mention the 4 students, Vivek, Pratik, Namvi, who probably also in the audience, Farah, they have won summer fellowship from St. Borrego Cancer Foundation since 2015. And from 2015 to 2018, every year I receive call from the foundation. And when they told me that my student won an award, that moment was very joyful. And I will never forget that. And very impressive work for those students. Last thing I want to say to the class 2018, do not be just a good doctor, be an outstanding doctor and an outstanding scientific researcher. Medical research from you guys will have more profound and important impact on patients. So thank you all for this. Best wishes to you all.
Thank you, Chief, for those great words. So now we go on to the other recipient this year of the award. Let me read a little bit about some of the comments we got from you, the nominators, about this person. They said, thanks largely in part to him, it is one of my career aspirations to become a physician scientist who would continue to contribute to the orthopedic literature over the course of my career. Another student said he encouraged me to apply to 6-year research tracks and provide a year of dedicated research. Another said the zeal and drive with which he approached his work were a model I hope to imitate, and we quickly began establishing a methodology and reaching out to the necessary contacts that would help facilitate robust experiments. This is for Dr. Jonathan Carmouche, who many of you know from orthopedic surgery, who is also the director of the Musculoskeletal Education and Research Center, or MERC, who's been very heavily involved not only with his own students working with him on projects, but with a lot of you throughout a number of other projects through the regular meetings and interactions. Dr. Carmouche has had about 11 students work with him so far, and several of those students already have publications that are out and more to come. So please join me in also honoring Dr. Jonathan Carmouche.
Thank you. Thank you, guys. Mission accomplished. I told Connor and Zach in the beginning, you make me famous, I'll make you famous. So here we are. This is what I consider fame. I want to use this forum for one early thing. So I talk to our guys a lot about integrity in research, integrity in what you publish, and I'd like to use this forum as an opportunity to confess. When I was starting my research fellowship, I had learned how to draw blood from a mouse from the guy who was outgoing, who had been in my position the year before. And the key is you hold the little cage, you pick them up in a certain way, and if you're relaxed, then they'll usually stay relaxed. And you can pick them up and then you draw the blood. So I don't like to fail publicly, so I came in that night and I started practicing. So I got this cage of mice. They were mixed sex, which is a problem. I reached in, I grabbed the mouse. I thought I was nice and relaxed, and then he turned around and gave me just a little nibble, and I threw him. And when I did that, he got stuck in the light fixture. So I was by myself. I freaked out. I climbed up on the desk. I knocked over the cage. So the bedding and the poop is everywhere, along with the other 5 mice. So I got 1 mouse up here. I got 5 mice running around. So I climbed up. I figured I got to get him because I don't know why. So I got him. When we wrote the paper, I included data from 6 mice, but I only actually found 4. So anyway, that's my confession. I feel much better after having said that. So I do want to say, the purpose of our group when we started that, it really was just sort of like a little idea. And I felt like I was kind of under-advised as a medical student, and I was under-advised as an undergraduate student. So I wanted to make it so that students who were interested in orthopedics could see what it's like to be an orthopedist, and also so that students headed towards a career, most of us it's mostly clinical, I wanted everybody to see that you can do a little bit of both, or you can dabble, and there is some meaningful contribution that we can make. So then how do you do that? Because I already was working more than I should. So equally selfishly, I figured out you can get medical students to do lots of stuff. So, and if you got a lot of medical students, you can get them to do even more. So that was the design of the group, was to help the clinicians in our group be academically productive, even though we're all busy clinicians, and also, of course, to help the guys and gals who are interested in going into orthopedics to have exposure, to see what it's like, to develop mentorships. The mentorship thing, to me, what I don't think I really understood, my goal was to give the guys coming behind me something more than what I had. And what I didn't really expect was just like what a blast it is, like how much fun it is to watch guys go from looking confused and like deer in the headlights, standing up here trying to present, and then making them do that every month, and then you see what the finished product is, as you guys will see or have seen over the course of the last several years. It's awesome. I mean, it's an unbelievable opportunity, and the fact that I get some sort of recognition for doing that is even more awesome. All of the stuff about motivating the students to go out and figure out how to answer questions, that's code for, again, you guys do the work. Like, we just sort of think together, and I kind of watch these guys come up with ideas and then watch them go pursue it. It's really an amazing experience. So I thank you guys for that. Congratulations and good luck.
Congratulations. Before I turn over to Dr. Laconte, if there are any animal rights organizations in the audience, disregard the earlier comments about the mouse in the ceiling. Sure, go right ahead.
I wasn't going to say that, but PETA is an unbelievable organization, and it did help me that they happened to have come through our lab. Seriously, this is not a joke, and they released a ton of animals. So the nice thing there is mine just sort of blended in with all the rest of them.
That was not scripted. Okay, we're now going to get on to more of the fun part of it and hear the presentations in the first section from the students who were recognized for their outstanding research projects. And I'm going to turn it over to Dr. Laconte, who will introduce the individual speakers. Listen.
So today we're going to hear from the students who earned Letters of Distinction in Research Scholarship. And Letters of Distinction in Research Scholarship are chosen based on students' manuscripts and oral presentations that happens between the years M3 and M4, so M3s, pay attention. Those Letters of Distinction are chosen based on the overall quality, significance, relevance, innovation, and clarity of the students' findings. And so we're going to hear from four students right now, and then we're going to take a break and go to our poster session. We're going to hear from another 4 students afterwards. Each presentation is going to be about 15 minutes, and then we're going to allow, sometimes it's the first time we've done this, allow time for about 5 minutes of questions, so enough time for 1 or 2 questions, and then moving on to the next presenter. So our first presenter to kick off the class of 2018 Letter of Distinction presentations is Dylan Cockrell. He worked with Rosalind Moran, formerly of the VTCRI, and he's going to talk to us today about exogenous insulin speeding neuronal hemodynamic coupling in hippocampal networks to enhance memory function. Dylan, how are you?
Sounds good. All right. Can you guys hear me okay? Yeah? All right, tuned in. All right, good. So as Dr. Laconte mentioned, I believe it was by virtue of having, excuse me, make sure this is turned on. Is this on? Okay. Virtue of having a last name early in the alphabetic order to have the privilege of being the first student to get to go today, which is awesome. Okay, so again, as she mentioned, I'm going to be talking to you essentially about intranasal insulin and how it affects memory networks. So the first thing I want to mention, I think when a lot of people hear insulin, naturally they think about its role in type 1 and type 2 diabetes, both in pathophysiology as well as in treatment. But there's a growing body of evidence in the importance of insulin and how it affects the brain. So briefly, speaking about normal CNS insulin functioning, we found that there are receptors localized both in the hippocampus and the entorhinal cortex, which are areas of the brain important for memory, as well as in the frontal cortex. And within the brain, it does similarly to how it does in the rest of the body in that it modulates glucose utilization. It's also been shown that insulin signaling is key for synaptic remodeling, and by that we mean transmission of signals from one neuron to the next. This is an important field of research, mostly because insulin pathophysiology has been implicated in Alzheimer's disease patients. And what we've seen is that Alzheimer's disease is characterized by both synaptic loss and memory impairment. Additionally, there's been shown to be reduced CSF insulin levels in patients with Alzheimer's, and as well as reduced brain insulin receptor sensitivity, similar to what we see in the rest of the body with diabetes. Additionally, it's also been shown there's an underutilization of both insulin and glucose that parallels the stages of cognitive impairment, both in Alzheimer's disease and in mild cognitive impairment. And this underutilization correlates with disease severity. So from there, I want to speak for a minute about why we're looking at this intranasal approach. First, there's been prior studies that have shown verbal memory improvement, both in healthy adults as well as in individuals with cognitive impairment, both mild cognitive impairment and Alzheimer's disease. And this has been shown both in single dosing of intranasal insulin as well as in regimented dosing over a period of 8 weeks. Additionally, the intranasal approach is important because it provides an avenue that is special compared to other approaches like intravenous or intramuscular. Specifically, when it's given intranasally, this allows the larger molecules of insulin to be delivered directly into the CNS through channels in both the olfactory and trigeminal epithelium. This is important because this nose-to-brain pathway is much less regulated by the blood-brain barrier. So if insulin, for example, was given intravenously or through the intramuscular, it'd be much less likely to be able to absorb efficiently into the brain. But when given it this route, we're able to have high rates of absorption. There's also been shown to be, again, significant higher CNS insulin levels compared to those other routes in prior studies. Importantly, the other key takeaway for intranasal insulin is there's been shown to be no change in peripheral plasma insulin or glucose values. So you have less concern for things like hypoglycemia compared to if you were giving this peripherally. Prior to this study, there were no fMRI studies that were examining memory and intranasal insulin, so that's where our study entered. So our big research question was, how does intranasal insulin administration affect brain activation and connectivity during memory encoding? So to answer this, we have a number of parts that we went through. Our first part was a blood draw pilot, which I'll talk about in a minute, that the IRB recommended just to ensure the safety of our participants. The second part, and the bulk of our study, was the fMRI portion where we employed a memory task and looked at the changes within the brain. And then from there, we used the results we got from that and did further analysis through an aspect of modeling known as dynamic causal modeling, which is a way of looking at specific brain regions and how they interact with each other. So our initial study, again, the blood draw pilot, our goal was to examine plasma insulin and glucose levels after both intranasal insulin and intranasal saline administration and determine if there were any adverse effects. Our design, we started with 8 participants. It was within subjects, so each person received both administrations, and we collected blood samples over an hour long. And you can see here that this is our device that we used to deliver the insulin and saline intranasally. So for our results, we found what we expected based on prior research, that there were no effects of intranasal insulin administration in plasma insulin or glucose levels after baseline correction. So for these charts, this here is the plasma insulin levels over time, with the yellow being the saline administration and the orange here being the insulin administration. And then over here, we have plasma glucose levels as well. Again, yellow being the saline and orange being the insulin. And there was no significant difference between the two. So for the second part of the study, again, this was the bulk. We started with 41 participants, excluded 1 after a scanner error. And this left us with 40 total that we split into insulin and saline groups. And this was a randomized, double-blinded part of the procedure. From there, we were able to match so that we had 10 insulin males and 10 insulin females, as well as 10 saline males and 10 saline females. And importantly, we also looked to see, based on BMI and pre-scan glucose levels, that the groups were equivalent as well. So again, there was no significant difference between the insulin group and the saline group based on BMI or pre-scan glucose here or here for saline. So for our actual task, first we started with an intranasal administration of either insulin or saline. Then each participant spent time in the fMRI scanner. And during their time in there, they viewed 180 images. And these were made up of 60 indoor scenes, 60 outdoor scenes, and 60 scrambled images. After they exited the scanner, we gave them an hour-long break, and then we had them do another task where they viewed 180 additional images, except this time the 60 scrambled images were replaced with a different 30 indoor and 30 outdoor images. And during this, we asked them to go through and identify each image as an image they had seen before or a new image. So we looked at the accuracy results before looking at the fMRI data, and we found there's no significant difference between the two. Both groups performed and accurately identifying each image in the low 70s, 70% for insulin and 73% for saline. So the bulk of our study from there was looking at data analysis. So I want to speak briefly about how fMRI data is analyzed initially. So the first thing I want to talk about is blood oxygen level dependent signaling, or BOLD signaling. And basically the way this functions is each person in the scanner is presented with some kind of stimulus. For our study, this was viewing different images of either indoor or outdoor scenes. That stimulus triggers localized neural firing to specific brain regions. And from there, that leads to localized increased blood flow And from that, we can detect bold changes. And again, this is measured in 3 dimensions across time in 2 seconds. So our first result we had was just looking at the effect of intranasal insulin. And what we saw, looking at these cross-section slices of the brain, was that there was a region in the frontal lobe right here you can see being tracked along. And this is the rectus gyrus when we mapped it out. And what we saw that this was significantly more activated for the intranasal insulin group compared to saline. So from there, we also wanted to look and see what was the main effect of images that were remembered compared to those that were forgotten for those individuals who received insulin. So again, these are additional activations that are being shown. And what we saw was, once again, you can see in this coronal slice, there's frontal lobe activation here, and as well as in this horizontal slice, this frontal lobe. So that was the rectus gyrus that we had identified previously, and again on a sagittal image here. But we also saw these big activations more in the temporal lobe, and you can see really well in this coronal view right here, these bilateral parahippocampus activations. So we knew from this initial data that we had 2 regions of the brain we wanted to focus on, that being the rectus gyrus and the frontal lobe, as well as the parahippocampus and the mesial temporal lobe. So from that data, we constructed what's known as a dynamic causal model. And what this is a method of analysis that takes the brain activations that we previously identified and tries to determine what their relationship is within each other. So our dynamic causal model, once again, focused on the rectus gyrus and the parahippocampus based on those prior bold activations. Additionally, in this model, we also included a balloon model, which is a hemodynamic model looking at blood flow and oxygen consumption. It's termed balloon model because it's based on increases in flow of blood flow that inflate a venous balloon with deoxygenated blood. So basically, it's looking to see as blood flow moves and oxygen is consumed, the changes within the brain. So this is our model here, and this is an image to look overall, seeing the rectus gyrus here in gray and the parahippocampus here. And each of these gray connections are neuronal connections that we wanted to focus on. So how are these 2 related to each other? What are the connections within the rectus that apply? And then what are the connections specifically within the parahippocampus that applies to itself as well? Then these balloon models were a way of looking at vascular effects. So again, arterial and venous here, both of the rectus for the balloon rectus and the balloon parahippocampus, looking at vascular changes there. So again, we wanted to look both at the neuronal changes and the vascular changes between these 2 brain regions. So our big table of results here breaks down all the results we saw, both for in red, these were our vascular effects that we saw, and then in gray, these are the neuronal effects, and we're going to break these down one by one. So the first neuronal parameter changes that we saw was we saw a stronger rectus gyrus intrinsic self-connection. And what we mean by that is improved autoregulatory inhibition. And so what that means is the rectus is essentially showing that in the insulin group, it's better able to focus itself and have stronger connections for that insulin group for remembered images. The main hemodynamic parameter change we saw was reduced rectus signal decay. So this is essentially an optimization of vascular flow within the rectus gyrus as well. So we had seen each of those parameters. So we have neuronal changes and hemodynamic changes, but we wanted to also know how did these correlate to the memory task as well. And so we saw strong positive correlations with the insulin group and memory performance for 3 specific parameters. Once again, this one here, which is the rectus, looking at hemodynamics, and again here with the parahippocampus looking at hemodynamics, and then finally a neuronal change that was focused on the parahippocampus as well. So our next question, again, looking at these parameters and how they influence memory, we wanted to chart out and see, based on the DCM network or our model that we constructed, what does that influence of memory look like? So what we used was these DCM parameters that we've just been talking about now here on the y-axis, and then performance on the memory task. So essentially going from left to right, these are improvements in their memory on the task. And what we saw was that this is a significant, strong positive correlation within the insulin group for how well they performed on the task and the strength of those parameters within the model. However, we didn't see this same effect in the saline group here. So there's essentially no significant correlation going on in the saline group. And then here are our significant regression coefficients and the actual details of their beta values as well that were significant. So what exactly does this mean? This is a lot of statistics, but to boil it down and break down what we are taking from this, what we're seeing is improved memory performance that's associated with, 1, a greater parahippocampal negative autoregulation, which is again a neuronal effect, and faster hemodynamic transmission through the vasculature surrounding the parahippocampal region. So again, a vascular effect there. But again, this is only for the insulin group, not for the saline group. So we have neuronal changes, we have hemodynamic changes or vascular changes, but what's the real culprit? Is there something going on here, one or the other, that's driving this? So this was our next step in our analysis. So first we did a group comparison where we grouped the hemodynamic changes or parameters together, and we grouped the neuronal parameters and compared the two. And what this does is this reduces the entire model where you're not looking at each one individually, but you're looking at these grouped together. And what we saw, again, so the gray ones here are the neuronal parameters, whereas the red are the hemodynamic parameters. And then this chart here is a breakdown showing which ones cross the threshold of being above 95 percentile. So again, the hemodynamic parameters for this study looking at grouping were the only ones, excuse me. Oh boy, there we go. So again, the hemodynamic parameters were the only ones that were significantly affected by the insulin, whereas there was no significance for the neuronal parameters here. So then from there, we also wanted to look at each individual participant as well, specifically within the insulin group, and further determine that we knew that this was really hemodynamic changes. And that was what we saw. So again, what this is a breakdown where each participant is singled out and allowed to and placed at the group average, which is this zeroing effect here. And then we looked at two models, one of which only accounted for neuronal parameter changes, and then another which only accounted for hemodynamic parameter changes. And what we saw was that each participant had a much better fit for the hemodynamic parameter changes compared to the neuronal. So every single participant, you can see that there are none north of this zeroing out line. Each one went south, preferring the hemodynamic model. So to summarize these results, once again, our pilot study showed no significant changes in peripheral plasma glucose or insulin as we expected. Our neuroimaging results showed that there were two regions of interest, the rectus gyrus and the perihippocampus, that were significant both for insulin and remembered images. And then for our DCM, or our model we constructed from those regions, we found that there were both neuronal and hemodynamic parameters that were significantly positive for the insulin group only. There was also a significant positive correlation between memory task performance and model strength, but only in the insulin group. And then once again, when we looked at group models and individual models, a hemodynamic parameter was preferred over the neuronal parameter for each participant. So for conclusions, our big research question was, how does insulin affect visual memory encoding? Again, we saw significant activation changes in both the frontal lobe with the rectus gyrus and in the perihippocampus. These changes were better explained by vascular compared to neural responses. And with intranasal insulin, there was better memory retention that was associated with faster vascular transmission and increased neuronal connectivity in the perihippocampus. So what's the broader impact? What we take away from this is that it appears that intranasal insulin accelerates neurovascular coupling, which allows for speeded encoding processes in memory circuits. This is important because it provides evidence for previously unknown mechanisms that have been demonstrated showing intranasal insulin and its memory behavioral effects, but we didn't know how it happened, and this provides some explanation for that. Most importantly, this is important context for future therapeutic approaches for mild cognitive impairment and Alzheimer's disease, two processes which before now have not had very successful approaches to therapy. So from that, I want to thank specifically Dr. Moran and Dr. Laconte, who have been very instrumental in helping me kind of piece all of this together and put this study to completion. And then Sarah Adams was a research coordinator in our lab who's been extremely helpful collecting data throughout the entire process. Cassandra Karam was also in the Montague lab who helped us with our blinding part of the procedure. And if I have any time left, I'll take questions and comments from the audience.
Time for a few questions.
You guys gonna take it easy on me? There's a hand back there. Yeah.
Yeah, so I had a question. So in your original study, you found that there was no difference in memory between that first hour. To actually find differences in memory, would you do a longer time period between?
So you're right, that we did not see a significant difference in the memory performance when just looking at the two groups. And we had a couple of theories for why that might be. One, the prior studies with intranasal insulin had been focused on verbal memory. And that's, when you think about the logistics of trying to do a verbal memory task in an fMRI scanner, is extremely difficult. We tried to fit our task that way, but it just wasn't going to work out. So this was specifically visual memory, which hasn't been studied before, so that could explain it. The other explanation is that these were healthy adults with presumably strong memory to begin with. Ideally, it would be great to do this study in healthy older adults or even patients who have memory impairment. But with the FDA approval, they wanted us to do it in patients who were not taking any additional medication, so that was our target population. What the changes that we saw were based on correlations with the actual fMRI data and the memory changes. So whether you could see that over time, I'm not sure, but with the verbal memory results that have been shown in prior studies, they did see positive changes over a period of 8 weeks, which was impressive. But there's also been research showing that you can see memory changes in just one single dosing with an hour later. All right, thank you guys.
I'm gonna let you find it. Okay, our next presenter is going to be Ashley Etchison. She worked, as many of you know, because I think you helped her out some, in the emergency department, and her talk is entitled Low-Dose Intravenous Ketamine for Treatment of Acute Migraine in the Emergency Department, and it was a randomized placebo-controlled trial.
All right, as Dr. Laconte mentioned, I studied low-dose intravenous ketamine for the treatment of acute migraine, and we were in the emergency department, and we were doing this through a randomized placebo-controlled trial. Before I begin, I'd like to briefly acknowledge my research team. It took many people to make this project completed. Especially like to thank my classmates Liam and Freddie and Vu Fan. Also want to thank the faculty and staff at the emergency department here at Carilion. And last but certainly not least, I'd like to thank all the consented patients that enrolled in our study. Without their willing participation, we would not have made the advancements that we did in this area of research. So to begin, I'd like to briefly discuss the clinical context of our research. So we were looking at migraine headache or acute headache in the ED. This is a very common headache disorder that we see in the US. About 1 in 7 Americans are affected annually, so perhaps you or someone you know is affected by a migraine. And according to 2010 government healthcare surveillance data, migraine or severe headache was considered the 4th leading cause of an ED visit. So not only is this condition frequent in the US population, but it also appears frequently in the emergency department. So this graph here is demonstrating a variety of trends in increase in prevalence and encounter rates in migraine headaches. And the top line, which is this dotted red line, with the steepest increase, that's actually referencing encounter rates in women, and that goes along nicely with the epidemiology of migraines and that women are more likely affected than men. So this slide is demonstrating a variety of acute migraine treatments utilized in the emergency department. This is a list of 8, but this is just 8 of many. So the point of this is to demonstrate that there is no current standard of care for acute migraine in the emergency department. In addition, each one of these medications is associated with a variety of contraindications, side effects, and even significant clinical windows. So for example, triptans must be given in the early course of a migraine, otherwise they lose their efficacy later on in treatment. In addition, there have been some studies on trends in migraine medication use in the ED. So this graph is demonstrating trends of use from 2001 to 2010. I'd like to point out the opioids category here on the left. So you'll see quite a big jump in that time period. And the reason I bring that up is because the US Headache Consortium currently doesn't recommend opioids as a first-line acute migraine treatment. So a combination of there being no standard of care for acute migraine with this increasing trend of non-first-line medications being used, those two things together bolstered our decision to pursue this investigational drug trial for acute migraine treatment. All right, so a little bit about the drug of study, ketamine. Its formal title is dissociative anesthetic. It is a non-competitive NMDA antagonist. So the NMDA receptor pictured here is a excitatory receptor in neurotransmission. It's involved in pain reception and pain modulation. And ketamine actually works as an antagonist, so it dampens the effects of this receptor in the body. So many healthcare providers know ketamine for its uses in procedural sedation or perhaps induction of anesthesia. And so typically for these types of medical procedures, ketamine is going to be used at a dissociative dose of at least 1 milligram per kilogram, but often greater. However, for effective pain management in a variety of clinical settings, ketamine is used at a subdissociative dose, which is considered less than 0.5. However, I'd like to bring up the point in red that in the emergency department for acute pain, a dose of less than 0.3 has been utilized in the literature. So for our study, we chose to look at 0.2. Ketamine also has a relatively stable cardiovascular profile, so providers are typically fairly comfortable giving this medication in low doses. And there are a few side effects that we want to watch out with ketamine. They are less common at these low doses, but it's still important to mention. So neuropsychiatric side effects are the group of side effects most common, or the ones to look out for, and that can include feelings of uneasiness or depression, hallucinations, also suicidality has been reported, and fatigue and dizziness. So these were things we were watching out for, but we expected them less so because of this low dose that we were utilizing. All right, so our research question. We were asking, can low-dose ketamine be utilized in the ED for treatment of acute migraine? And we had two hypotheses. The first one being that ketamine-treated subjects would exhibit superior pain score reduction compared to placebo. And the second part was that ketamine would demonstrate an adequate safety profile. All right, so for our methods, we kind of had a three-tier approach. The first portion of the study was subject identification and recruitment. So for this portion, whoever was assigned at the time to enroll patients was monitoring EPIC, or our electronic medical record. They were looking at patients in the ED in real time, and they were trying to see if there were any patients in the waiting room with migraine or headache. So if patients met our initial age criteria, had a chief complaint of headache, and then met the ICHD criteria, which stands for International Classification of Headache Disorder, then they were enrolled in our study. So I want to expand briefly on that criteria. So this is a standardized criteria that's used in most migraine trials, so we wanted to stay consistent with the literature. And each type of migraine has a code associated with it. So the migraine that we actually saw most common in our patients was this first one, migraine without aura. So I'll expand on that. So for each research subject that was recruited, they had to be asked a series of questions. And one of the first questions they were asked is, what kind of migraine do they have? So we were using this checklist to make sure our patients had an official or typical migraine according to these codes. So like I said, migraine without aura was our most commonly seen migraine. But our second most commonly seen migraine was migraine with aura, so the 1.2 category. And to have a migraine with aura, a patient has to have those same characteristics that I just showed, plus they have to comment on the fact that they had an aura, which is basically a visual disturbance just prior to the onset of migraine pain. Most commonly, it is a visual disturbance, so patients might see bright flashing lights or zigzag lights, but that is technically considered an aura for this study. All right, so after our patients were consented, they were randomized to one of two treatment arms. This was a double-blinded study, so the patients, the healthcare personnel, nor the research investigators were aware of the treatment given to the patient. Before the intervention was given, demographics and baseline pain data were collected from the patients, and then the patients were given one of two treatments, either 0.2 milligrams per kilogram of IV ketamine or equivalent volume of normal saline. And then the research investigator returned to bedside at 30 and 60 minutes to collect a variety of data points. The most important data point, which was our primary outcome, was the NRS 11 pain score. And this is a verbal pain scale that we would ask our patients from 0 to 10, how are you feeling? So from 0 means they have no pain, 10 means they have the worst pain of their life. So that was our primary data point. In addition, we also collected categorical pain scores, so is a patient's headache mild, moderate, or severe. We also looked at functional disability, how impaired was this patient from their headache? Were they mildly, moderately, or severely impaired? We also monitored side effects. And at 30 minutes, we asked patients if they would like rescue medication. And the patient was asked at 30 minutes, and then if they said yes, then we immediately notified the attending physician, and the attending physician gave that patient rescue medication based on his or her discretion. So that was not a standardized component of our protocol. And then finally, we asked patients at 60 minutes at the conclusion of our study if they were satisfied with their treatment. Okay, so this schematic shows the flow of enrollment. We enrolled for 12 months from March of 2016 to March of 2017. In total, we assessed 173 patients for eligibility. And out of that number, 138 were not included in our study. The vast majority of these patients weren't included because they did not meet some aspect of our inclusion or exclusion criteria. So that was the 92 patients here. Most commonly, a reason for not meeting inclusion criteria was that stringent classification criteria that we had all our patients go through. So a lot of patients didn't meet all those requirements, so they weren't included in the study. One number that I want to point out that we were pleased to see was the decline to participate. So you'll see that number is 26, which with all things considered, I think it's pretty good. When we started our study, we were pretty concerned that with this placebo arm that we might have a lot of pushback from patients, and they might not want to be enrolled in a study where they're currently in pain and there's a chance they might receive saline placebo. However, we did not encounter that problem very frequently, and we were very excited to see that the community was very receptive of our research. So from that 173 patients or subjects, we were able to get 35 enrolled, 17 in the ketamine group and 18 in the placebo group. And this table is demonstrating the baseline demographics of our patients. So the majority of our patients were female. On average, they were 35 years old. 50% of them had a category of 1.1, which is that migraine without aura. And then a quarter of them had 1.2, which is migraine with aura. And on average, our patients presented with an 8 out of 10 pain score, and 77% commented they had a severe headache. All right, so this slide walks through the primary outcome of our research. And like I said, we're utilizing that verbal pain score number. And we have a couple important timelines. So we're looking at the reduction in pain score from 30 minutes after medication administration from baseline. So we're going to find that for each group, and then we're going to take the between group difference, which is going to be in this box here, and like I said, that is our primary outcome and our most important data point. So for the ketamine treated group, after 30 minutes, there was an average pain score reduction of 1.97, and for placebo group, it was 2.06. So for this box right here, we were expecting a few different things. First of all, we were expecting a positive number, because that means that ketamine would have outperformed or had a greater reduction in pain compared to placebo. We were also expecting a clinically significant value, which in literature is a value of at least 1.3. And last but not least, we were hoping to see statistical significance. So the number we calculated was negative 0.09, which, like I said, is our primary outcome. So what this number tells us a few things. One, it's negative, so it means that placebo slightly outperformed ketamine, and it's also lower than 1.3, so it's not of clinical significance. And thirdly, it's not statistically significant. So this value here tells us that ketamine was not superior to placebo in terms of pain reduction for acute migraine patients. We also completed these values for the 60-minute time period, but like I mentioned, at this time period, many patients had received rescue medication, and like I said, it was not standardized, so we can't include that as a primary outcome. We were mostly focused on this 30-minute mark. So in conclusion from this slide, we didn't see any significant differences between ketamine or placebo groups at 30 or 60 minutes, but we did see statistically and clinically significant pain score reduction within each group at 30 and 60 minutes. So these asterisks here are denoting that these values are statistically significant. So within each group, we did see some significance, but the study was powered, and the primary outcome, of course, was the between group difference. All right, so this graph is demonstrating the overall trends in pain score reduction. So the blue line is representing our entire cohort. So that entire cohort, in addition to the individual groups, did show an overall downward trend. So patients were improving in their pain scores. And we also collected a few other data points I mentioned. So for categorical pain, that mild, moderate, or severe headache, we didn't see any significant differences in terms of reporting either an improved or unchanged headache between both groups. And we saw the same result for functional disability. So both groups performed similarly in terms of improved or unchanged functional disability. We also monitored side effects. We used the SIRSDA side effects model, and we looked at 9 different side effects. We asked patients about these at baseline, 30 and 60 minutes. And in 2 of them, ketamine patients rated higher values than placebo. And that was in generalized discomfort at 30 and 60 minutes, as well as fatigue at 60 minutes. Otherwise, the side effects performed similarly in both groups. At 30 minutes, like I mentioned, we asked our patients if they wanted rescue medication, and we didn't find any significant differences between the 2 groups in terms of rates of asking for that medication. And our last comparison was treatment satisfaction at 60 minutes, and we did not find any significant differences between these 2 groups. So both ketamine and placebo groups reported similar rates of satisfaction. All right, so in terms of our conclusions, so we think this these results are significant, and we think they can certainly add to the body of literature for acute pain in the ED. And this is important because this was the first placebo-controlled trial investigating low-dose ketamine for acute migraine in the ED. So we hadn't seen this anywhere else in the literature. That's why we were excited to at least take a stab at it. And so for our overall conclusion, we determined that 0.2 milligrams per kilogram of IV ketamine did not produce a greater pain reduction compared to placebo. However, ketamine did demonstrate an acceptable safety profile. So our ketamine-treated patients did not report any serious adverse events, nor did they report any serious neuropsychiatric side effects. And interestingly, the only patients who reported hallucinations were those treated with placebo. So we do have a few considerations for future studies. We think this data can serve as a springboard for future studies. So one consideration would be to increase the dose. So current literature, Lee et al. Published a great review saying that the optimal dose for acute pain treatment in the ED is 0.3 milligrams per kilogram, and we used 0.2, so it's a consideration to up the dose. And we could also consider a different route of administration. So for our study, we used a single one-time bolus via IV. However, in the literature, there's been a few reports of infusions being quite effective, not only at reaching pain relief faster, but also reducing side effect severity. So before I close, I'd like to just briefly mention a few places I was able to present this research. I was very fortunate to go to Athens, Greece with Leah. We presented at the 11th European Congress of Emergency Medicine back in September. And then also last year in October, I went to DC and presented at the Scientific Assembly at the American College of Emergency Physicians. And are there any questions? Yes.
You assembled your patient population. Did you do any screening that looked at how well controlled the response to the migraines had been prior to your research?
In terms of like other treatments they've tried?
Right.
That's a really good question, and we did not ask that. Overall, we saw people had taken a variety of treatments. Some patients even came in and knew what they wanted. So I think it's just all across the board, and it speaks to that there is no standard of care. So each patient is unique in the way they respond to their medications. But it's a good point. We didn't ask that.
Sort of similar line of thinking. They were all assuming not on any form of prophylaxis for the migraine?
Correct. We would ask each patient before they came or when they came in what previous medications they'd taken, and we wanted to make sure that there was a treatment-free window. Mm-hmm. Anyone?
Okay, so our third presenter of the session will be Pratik Kanabur. You already heard sort of a brief intro to his research when Dr. Shang was talking about his award. He's going to talk about patient-derived glioblastoma stem cells and their differential response to targeted therapies.
Put my phone back. Thank you. Oh, that's fine. Thank you. All right. Good afternoon, guys. The title of my presentation is using various treatment modality against glioblastoma stem cells. My mentor is Dr. Shang at BTCRI. So a little bit of background. Glioblastoma is the most common malignancy of the central nervous system. It's a grade 4 astrocytoma, meaning it's derived from the astrocyte cells of the brain, and it's highly lethal and highly malignant. In fact, the 5-year survival time is less than 5%. And the median survival time, despite decades of research, is only 14.6 months. So I want to highlight the urgent need for more efficacious treatments for this terrible disease. So specifically kind of talking about glioblastoma stem cells and conventional treatment. So here's a depiction of the tumor, the stem cells kind of intercalated among the tumor. And conventional treatment involves surgical resection, it involves radiation therapy,
and chemotherapy with the agent temozolomide. However, even after all this, the stem cells that are resistant to this treatment still remain. And these stem cells are able to replicate and differentiate, and they cause the tumor to regrow. And if this tumor regrows, the glioblastoma recurs, and you see this on the CT scan a year later. And at this point, the patient has no more treatment options, and it leads to their mortality. However, consider an alternative treatment for these involving glioblastoma stem cells. So the same depiction of the tumor, however, we're going to target the stem cells first. So using a stem cell targeted treatment, you destroy the stem cells, and you take away the opportunity for them to grow and differentiate again, and you're just left with the tumor. And then you hit it with chemo, radiation, and surgery, and it leads to the regression of the tumor. However, before treating these cells, we have to understand kind of what glioblastoma stem cells are. So let's take a step back and first consider what stem cells originally are. So stem cells have two important properties. The first is that they're able to self-renew, or that one stem cell is able to make multiple different stem cells. The second characteristic is they're able to differentiate. So a stem cell can become a more mature cell lineage. So here, a neuronal stem cell is becoming a glial cell and a neuronal cell. However, if these stem cells pick up a mutation, they become a glioblastoma stem cell. In addition to self-renewal and differentiation capability, they're also able to replicate in a way that they have different patterns of replication and different methods of replication. That kind of you can see in different tumor growth and different patterns of growth. And next, they have tumorigenic potential in that they're able to initiate and form the tumor. And lastly, these stem cells, because they're resistant to most therapeutics, they have resistant to common drugs. So our research question is devise effective strategies to combat therapeutic drug resistance. Do glioblastoma stem cells isolated from different patients have different properties? And do glioblastoma stem cells from different patients respond to treatment differently? So our first goal was to isolate these stem cells from patient samples. So in collaboration with Dr. Gary Simmons and the rest of the neurosurgery colleagues, we isolated, or we consented patients that are undergoing surgical resection for their glioblastoma, and the patient samples were given to our lab, and the tumor was digested into single cells, and these single cells were cultured in a serum-free stem cell media for a period of time until they're able to form spheres. So here's some examples of some of the Carillion patient spheres that we have. So here's VTC34, VTC36, 56, and 64. And even though they just look like similar, like in terms of blobs, they all have different characteristics that I'll continue to describe in my talk. So the first is self-renewal. So again, remember, self-renewal is the ability for one stem cell to become multiple stem cells. And the way we tested that was using a self-renewal assay where we took these tumors, we digested them into single cells, and we put them into a 96-well plate ranging from a density of 256 cells per well down to one cell in each well. And what we observed was the formation of spheres. So here we can see a sphere of about 32 cells per well, and here a little bit smaller, 16 cells per well. And what's kind of important to know is that if it's only able to form a sphere at these high concentrations, that makes it less stem cell-like. Versus if it forms a sphere at a higher or a lower concentration, it is more stem cell-like. It's a little bit backwards. So we plotted the percentage of wells without spheres against the number of cells plated. And using this graph, and from the x-intercept, we determined the sphere formation number, or the number of cells required to form one sphere per well. And here in this table here, we have some established cell lines, and we also have these Carillion patient cell lines as well. And again, it's important to remember that the sphere formation number, the lower the number, the more stem cell-like it is. So here, for example, VTC36 is more stem cell-like than VTC34. So next, we diluted these cells down to one cell per well, and we want to see what one cell looks like when it's dividing, to see if they have a different pattern of growth and division. So we used an established cell line, GBM10, and we also used two Carillion patient cell lines, VTC64 and 34. And as you can see here in single cell video format, these cells are dividing in a different fashion and they look like they're different. The next thing we did was we looked for certain markers that these cell lines express. So here is a Western blot looking at all different kinds of patient samples and established cell lines, seeing for certain markers. The first marker that we looked for was the stem cell marker nestin. And as you can see, these express higher levels of the stem cell marker nestin and lower levels of the differentiation marker glial fibrillary acidic protein, or GFAP, which is a marker of differentiation as a mature cell line. And they express lower levels, as you can see. So the next thing we looked for was tumorigenic potential, whether that these stem cells can form a tumor in a mouse. And we compared two different patient cell lines, VTC36 and VTC37. VTC36 was nestin positive and GFAP negative, or VTC37 was nestin and GFAP positive, meaning it had more of a differentiated cell population. And we injected the same concentration of cells subcutaneously into the mouse flank, and we observed for the growth of a tumor. So here you can see the tumor in the mouse. And we found that VTC36 formed a tumor, but VTC37 did not at the same concentration. And we took out that tumor and we sliced it up and looked at it under H&E staining, and you can see that it clearly looks like a tumor and it resembles the same histological features of glioblastoma. So we characterize these cells in terms of self-renewal, their markers, what a single cell looks like, and if they're able to form a tumor in a mouse. So now we want to see if we can target these cells, if we can use drugs to eradicate them. And so remember, these stem cells are resistant to conventional therapy. And so our aim was to target these stem cells and see if they are susceptible to treatment. So amongst the many drugs that we use in lab, two that I'm going to focus on in this talk are imatinib, which is a platelet-derived growth factor receptor alpha blocker, so a PDGFRA blocker. So it blocks this receptor and it prevents PDGF from working, and which releases signals of proliferation and survival. And the next one is a gamma secretase inhibitor 9, which prevents the gamma secretase cleavage of the Notch receptor, and it prevents Notch from doing its signaling pathway, which also is implicated in proliferation and survival. So from now on, I'm going to call imatinib a PDGFRA inhibitor, and the gamma secretase inhibitor is a Notch inhibitor, just to make it easier. So we wanted to see what these drugs did in terms of when we treated them cells. So we used two established cell lines, U251 and GBM10, and we treated them with the same concentration of drug. And here we can see that the U251, the PDGFRA inhibitor had a significant effect, but the Notch inhibitor did not seem to work. Whereas with GBM10, the PDGFRA inhibitor did not work, but the Notch inhibitor did. So that led us to wonder whether these cells express different markers, as if they don't have the target for these drugs, they're probably not going to work as well. So what we did was a Western blot to look for if they have markers, and we used all the different kinds of patient lines and established cell lines. So specifically focusing here on this, you can see maybe that U251 has some expression levels of PDGFRA, but no Notch, versus GBM10, which expresses Notch, but no PDGFRA. So looking back at the same picture here, we see U251, which was responsive to PDGFRA, but not Notch. And it was positive for the receptor for PDGFRA, but negative for Notch. Where GBM10 was the opposite, and we see the opposite in terms of the marker. So it doesn't have PDGFRA, but it does have the Notch receptor. So we also continued these experiments on the Curlian patient cell lines. So here we can see VTC36, which had both PDGFRA and Notch, and you can see that there's a clear effect of these drugs, and they're inhibiting these cells from growing. We quantify these with a similar self-renewal assay where we put 200 cells per well down to 1 cell per well, and we wanted to see if these drugs would inhibit sphere formation and if that was possible to calculate. And what we saw was that the drugs did indeed inhibit sphere formation. So again, remember, the smaller the number, the more stem cell-like it is, Or in this case, the larger the number, that means that this drug is inhibiting the spheres from forming. So specifically looking at VTC56, the PDGFRA inhibitor had a greater effect than the Notch inhibitor, and that correlates perfectly because the PDGFRA level was high, and the Notch level was still there, but it was low. We also quantitated this in another way using a viability assay, using an MTS colorimetric assay with VTC37, and we found that the PDGFRA inhibitor had a significant effect on decreasing viability, and the Notch inhibitor, which decreased viability but was not significant. And we correlated that with the results, and you can see that PDGFRA had a medium expression level and Notch was low. Thus, it makes sense that this inhibitor was more impactful than the Notch inhibitor. So we were able to characterize these stem cells. We were able to see that we can develop markers and treat them specifically. However, all this was done in a 2D culture. And we wanted to move these experiments to more of a 3-dimensional way. So what we decided to do was this new kind of way to look at a 3D culture with an organoid. So what an organoid is are single cells combined with a matrigel or like a 3D matrix, and they're combined together to form a patty. And this patty is pipetted into a well, and we observe for the formation of an organoid. And what it looks like is here in a petri dish, this is an organoid. It's almost like a little tumor growing in a dish. And what's really cool about the organoid is you can not only see microscopically what's happening, but you can see grossly what's happening here too. So here we can see the sphere forming, and this is kind of what it looks like grossly. And as you continue to watch the growth, you can see the spheres are getting bigger, but in addition, this organoid is changing. And by 6 weeks, you can clearly see that there's a significant change. So we wanted to see if we could see a change with drug treatment. So we used a PDGFRA inhibitor against the treatment of organoids. And we compared VTC36, which had that receptor, so we were anticipating that this drug would work. And we compared to VT64, which did not have this receptor. And what we found, looking at the organoids, they had different shapes. And once we treated them with the drug, you can see here that there's some change in the organoid. And by 4 weeks, this organoid had been destroyed, while VT64 looked pretty similar, if not better. So summary and conclusions, glioblastoma stem cells can be isolated from patient tumors. These tumors have different stem cell properties, including self-renewal, what single cell formation looks like, if they're able to form a tumor in mice, and their markers. They can be targeted for treatment using specific receptors, and they have different patterns of drug susceptibility. And they can be used to form 3D organoids that serve as a more realistic model for glioblastoma drug treatment experiments. Thank you so much. I'd like to acknowledge everyone in the Shang Lab, the fellowship support from these various organizations that donated a lot of money to my research. And last and most importantly, I'd like to thank all the patients. I'd like to thank the patients that donated their samples to my research, and the patients that I was able to shadow in the clinic and see, who are very excited and thankful for the work that we're doing. And I think we all do our work for the patients, and I'm glad I got to be a part of it. Thank you.
Time for some questions.
Oh, yeah.
Did you look at any other potential breakthrough stem cell markers, or consider looking at any other ones?
Yeah, we considered looking at CD133 and Notch and Nestin and SOX9 and a lot of other ones, and they had various kind of results. Like, these stem cells look different, and I think for publication purposes we chose to do this one because it kind of correlated really well with our results. But yeah, I mean, people use different things, and this whole stem cell idea is almost kind of a mythical concept. So they all have different kind of markers that people agree or don't agree on what it has. Well, temozolomide crosses the drug brain, but I didn't talk about that. Imatinib and the gamma secretase inhibitor, I believe, cross the blood-brain barrier, but that is definitely a concern. that these drugs in general are not crossing the blood-brain barrier very well. So that's obviously a consideration. We had ideas of like opening up the mouse brain and injecting the mouse, or like electroporating to get a blood-brain barrier, but that is just a general concern for this disease that you can't penetrate very well.
So our last presenter for this session will be Vu Fan. He worked with Jamie Smith in VTCRI. Today, Vu is going to talk about the disruption of cardiac gap junctions during adenoviral infection and the implications for arrhythmogenesis.
Thanks. So let's see, good afternoon, everybody. Today my presentation is going to be on the disruption of cardiac gap junctions during adenoviral infection and its implications for arrhythmogenesis. And thanks to the committee for allowing me to present today as well. And so to start, I'd like to provide context for the work that we do by discussing heart disease. It is the leading cause of death among individuals in the United States and worldwide. More specifically, up to 20% of all cases of sudden death in young adults is due to the development of myocarditis, which is simply an infection of the heart muscle. However, all forms of heart disease, whether it be ischemic or non-ischemic, and our work focuses on the viral etiologies of heart disease, are associated with an increased risk of arrhythmias and sudden cardiac death. And so key to the understanding of arrhythmias is the concept that the heart is fundamentally an electrical organ. And so in order for the heart to pump properly, there has to be coordinated contractions of the ventricles, which is accomplished through the literal transfer of an electrical current through the myocardium by the organized propagation of an action potential through the heart. And so when you think about that on a cellular level, what simply has to happen is effective intercellular communication. More specifically, ions have to be able to flow between adjacent cells in order to transfer that electrical current properly. And so when we think about arrhythmias, which are fundamentally dysfunctions of the normal electrical circuitry of the heart, we can also think of them as disruptions in intercellular communication as well. And so key to this idea of intercellular communication is the idea of the gap junction. These are sets of proteins that serve to connect the cytoplasms of adjacent cells. They are involved in everything from electrical to immunologic to metabolic coupling, and they can have non-junctional roles as well, including cell cycle regulation. Now, gap junctions are made up of a protein called connexin. We study primarily connexin 43, which can have a secondary role as a tumor suppressor as well. And so in the connexin life cycle, 6 connexins will oligomerize to form what is called a connexon, which is shuttled to the cell surface, at which point connexons from adjacent cells can actually bind to form what is called the gap junction channel. And they function by allowing the passage of small molecules up to 1 kilodalton in size. And so in the case of directly coupling adjacent cardiomyocytes, in order to transfer a current, small ions such as potassium may flow in between cells. In the case of immunological coupling, secondary messengers may flow between cells in order to initiate an immune response as well. And so not only is connexin crucial for normal cardiac function, but it's also been shown that there's significant remodeling of connexin 43 expression in the failing human heart as well. And so in this immunofluorescence study, which allows us to visualize the position of specific proteins, the position of end-cadherin, which indicates the position of the cell junction, has been stained for in red. You can see the position of the cell surface in white for ease of sight, and the position of connexin 43 has been stained for in green. And so when we take a closer look, you can actually see in the failing cardiac tissue that there's significant failure for that connexin 43 to localize properly to the position of the cell junction. And so there are many forms of potential heart failure, but the kind of potential heart failure that we seek to study is called viral myocarditis, more specifically adenoviral myocarditis. Now, in this study looking at samples of cardiac tissue from 239 patients diagnosed with viral myocarditis, adenovirus was actually found to be the leading pathogen present in over half of all samples at 55%. In the summer of 2005 in Havana City, Cuba, there was actually an adenoviral myocarditis outbreak leading to 8 pediatric fatalities associated with cardiac failure in the setting of adenoviral myocarditis as well, indicating that not only is adenovirus a major cause of myocarditis, but also this is a disease with real-world implications as well. And so when you think about sort of the typical clinical presentation of this virus, most of the time this will just present as simple respiratory infections that approach, a bronchitis or a pneumonia. However, in those who are young and those who are immunocompromised, you can have more serious infections as well, in particular infections of the cardiac tissue. And so these patients may present with arrhythmias that actually resemble myocardial infarctions or heart attacks, and if left untreated can lead to the development of acute heart failure and potentially fatal arrhythmias as well. When thinking about this on a molecular level, the adenovirus is an icosahedral, non-enveloped, double-stranded DNA virus. It is typically contracted through close contact or inhalation. And once inside the cell, the virus has to be able to do a few things in order to be able to achieve successful infection. And so as a double-stranded DNA virus, it has to be able to uncouple key cell cycle checkpoints in order to push the host cell into S phase, which is the phase of the cell cycle responsible for DNA replication, in order to eventually replicate its own DNA or its viral DNA. And all while doing so, the virus has to be able to inhibit the host cell immune response as well. When thinking about the adenovirus genome, you can generally separate it into two categories. The early genes, and part of their function is to push the host cell into cell cycle entry, versus the late genes, which tend to code for more structural proteins, an example being the hexon protein of the adenovirus. And so in terms of the scientific premise of our research, remember before that we said, connexin can serve as a gap junction, but it can also have secondary roles as well. And so it's actually known that intracellular communication actually suppresses cell cycle entry. And so if adenovirus were able to downregulate connexin 43 expression, it would more easily be able to push the host cell into S phase, initiating eventual genome replication. It's also known that connexin 43 helps propagate the initial immune response to viral infections, in particular through the flow of secondary messengers that allow for interferon secretion, which actually helps fight viruses. And finally, connexin 43 is known to play a key role with respect to antigen presentation in terms of the transfer of key viral antigens and the adaptive immune response as well, which is a later immune response that's more tailored to specific types of pathogens. And so if adenovirus were able to downregulate connexin 43 expression, it would help blunt these immune responses as well. And so in terms of the significance of our research, there's actually no literature out there establishing a connection between wild-type adenovirus infections and its ability to dysregulate connexin 43 expression. So ours would be the first work to do so. In addition, we would be able to learn more about adenovirus pathophysiology, as well as connexin 43 dysregulation in states of disease. And so with that in mind, our general research questions is what are the effects of adenoviral infection on connexin 43 expression and gap junction formation? 2, what viral proteins are sufficient to affect connexin 43 expression? And finally, does connexin 43 affect viral replication through junction-dependent or independent functions? And so to get at this first question, we hypothesized that given that connexin 43 can limit the cell cycle and enhance the intrinsic antiviral immune response, that early adenoviral proteins actually target connexin 43 expression and function to facilitate viral genome replication. And so for this first experiment, what I did was I actually infected cells with adenovirus and analyzed for connexin 43 protein expression through Western blot. And we saw over the course of 48 hours that there were actually significant decreases in connexin 43 expression. When quantified and with subsequent statistical analysis, those changes were actually found to be significant. And so that occurs at the level of protein. Next, we wanted to determine what happens at the level of transcription. And we did so through RT-qPCR. And we also saw that with the onset of infection that there were decreases in GJ1 or connexin 43 mRNA expression as well. And so next we wanted to determine what happens to localization through an immunofluorescence experiment. We stained for the position of the cell membrane through WGA stain, which we can see here. We've confirmed for the presence of successful infection through a stain for the hexon protein, which we can see in red, appropriately in our infected cells and absent in our control cells. And finally, we've stained for connexin 43 in green. And so when we take a closer look at connexin 43, we see abundant expression in our control cells and a significant decrease in our infected cells. And so given that information, the second question we wanted to answer, what viral proteins are sufficient to affect connexin 43 expression? Now, there's actually previous literature showing that the E4 gene of adenovirus was capable of significantly downregulating connexin 43 expression over 4 days through the mTOR growth signaling pathway. This mTOR growth signaling pathway is actually present in the host cell, and it's actually hijacked by the virus. And when initiated, it can actually allow for the translation of specific growth mRNAs, leading to eventual viral replication. However, when you look at the E4 gene, it actually codes for 7 different proteins. And so through research, we actually saw that E4F1 and E4F4 were actually capable of stimulating this mTOR growth signaling pathway. And so with that information, we sought to answer what viral proteins are sufficient to affect connexin 43 expression. We hypothesized that since E4F1 has been shown to activate the mTOR growth factor signaling pathway, that ORF1 would be sufficient for downregulation of connexin 43 expression. And so to get at the second question, I actually created plasmids that ectopically express our proteins of interest, either ORF1 or ORF4, with confirmation of expression through the use of an N-terminal hemagglutinin tag, which we could confirm the presence of. And what we saw was that with successful expression of ORF1, that there were significant decreases in connexin 43 expression. When quantified with subsequent statistical analysis, those changes were actually found to be significant as well. Next, we wanted to determine what happens at the level of localization. And so here we see a stain for our HA tag. Here, this confirms successful expression of our control plasmids, whereas here, this confirms successful expression of our ORF1 protein. We see our stain for HA here in red, connexin 43 in green. And so if you take a look over here at our cells that are expressing HA ORF1 in red, when we take a closer look at connexin 43, we see that with successful expression of ORF1, significant decreases in connexin 43 expression as well. And when quantified, those changes were also found to be significant compared to our non-transfected cells. And so finally, the last general question we wanted to get at was, how does connexin 43 expression affect the ability of adenovirus to replicate? And we did so through the use of plasmids that ectopically overexpress connexin 43 and analyzed for protein expression and genome replication. And so what we saw was that with overexpression of connexin 43, significant decreases in hexon expression, which you should remember is that structural protein of the adenovirus. Next, we completed RT-qPCR to see the effect on genome replication. And we saw that with overexpression of connexin 43, significant decreases in adenoviral genome replication as well. Next, we wanted to look at the inverse. Last time we increased connexin 43 expression, what happens now when we decrease connexin 43 expression? And we did so through the use of siRNA-mediated knockdown of connexin 43. And we next looked at the effects. And so we saw that when we decreased connexin 43 expression, we actually increased hexon expression. Similarly, with decreases in connexin 43 expression, there were also increases in adenoviral genome replication as well. And so with this in mind, we wanted to answer our last research question of, does connexin 43 affect viral replication through junction-dependent or independent functions? Remember, connexin 43 can act as a gap junction, but it can also have secondary roles as well. And so we were able to get at that question through the use of connexin 43 mutants, which actually express connexin 43, but express gap junctions that are non-functional. And so we utilized R2OTE, which expresses connexin 43 but is trapped within the cell. And we also utilized a plasmid called T154A, which expresses gap junctions, which localize to the surface but remain closed. And so what we saw was that when we introduced these dominant negative plasmids with subsequent infection by adenovirus, that we were actually able to rescue hexon expression. And so this is important because this actually implicates the gap junction functions of the protein in terms of its ability to suppress adenoviral infection. And so for our last experiment, thus far everything has been completed in respiratory epithelial cells. And so if you disturb gap junctions in the respiratory epithelium, the typical presentation will be cough or pneumonia. However, in the case of adenoviral myocarditis, it can lead to the development of a potentially fatal arrhythmia. And so for our last experiment, we actually completed them in a cell line called induced pluripotent stem cell cardiomyocytes, which are basically stem cells that have been converted to cardiomyocytes. And we saw that with the onset of infection that there were decreases in connexin 43 expression as well. And so lastly, what are our conclusions? We actually saw that with infection by adenovirus, more specifically expression of the E4 ORF1 protein of adenovirus, that it was capable of significantly downregulating connexin 43 expression through transcription. Next, we saw that with overexpression of connexin 43, that it actually suppressed adenoviral genome replication and protein expression. And that with the introduction of dominant negative plasmids that actually form gap junctions that are closed but localized to the cell surface, that we were actually able to rescue adenoviral protein expression, which implicates the gap junctional qualities of the protein in terms of its ability to suppress viral infection. And so in terms of where we want to go for future work, we'd want to assess the arrhythmogenic potential of infection, again, in induced pluripotent stem cell cardiomyocytes. Interrogate more the mechanism of how E4F1 works in terms of connexin 43 expression, and finally look at how gap junction intercellular communication suppresses adenoviral replication. Before I forget, I'd like to thank everybody in my lab for all their help in supporting me throughout this process. Patrick Calhoun, my co-author, my mentor, Jamie Smith, Mike Seitz, Chris James, and everybody else in the lab as well for all their help and support. And so that is it, and I'm ready for questions.
Okay, some questions for who?
Thanks, Al.
Okay, so that concludes our first oral session. I want to remind the students that you all received a link for a survey in your Carilion Clinic email, so you have to check the Carilion Clinic email. That survey is for you to go look at the posters and select student awards. So select the most tweetable poster, select the most clinically impactful poster, those sorts of things. So vote for three in each category. The voting closes at 3:30, and then we will award those at the end of the oral session that starts at 3:30. There are refreshments down in the cafe, and go see your even number posters now.
So yeah, presenting.